Very good day, everyone. My name is SHAHARA ZULFAKAR, and today I would like to present to you the findings from our scoping review study, which is on the occurrence of antibiotic resistance in aquaculture products and aquaculture environment in Malaysia. As an overview, aquaculture and fishery sectors is one of the fast-growing food sectors in Malaysia. The increased demand from consumers contributes to greater usage of antibiotics in promoting the growth of red species and to treat any bacterial infection diseases in the aquaculture products. However, misuse of antibiotics usage has led to alarming rate of resistance among bacteria towards antibiotics. And therefore, this has led to the emergence of widespread antibiotic-resistant bacteria, which is one of the main public threat to human health whole worldwide and is affecting the safety and quality of the seafood product. Therefore, this study aims to report the overall pattern of antibiotic resistance in aquaculture products and its environment in Malaysia, and also to identify the type of aquaculture products and its environment that are associated with antibiotic resistance in Malaysia. A scoping review approach is adopted in this study for identifying, screening, and select the relevant articles for this study. 3 online databases are being used to search for the articles, which are PubMed, Scopus, and EBSCOhost, and we end up with 33 final articles that we extracted the data and we analyzed for this scoping review. Overall, we found that the common method used to isolate the bacterial strains are by using plate culture method in combined with PCR to identify the species. And it is not surprising to see that the antibiotic susceptibility test method that are being used is by using this diffusion method. Overall, about 80% of the articles are targeted only on one specific targeted bacteria, which are commonly Vibrio parahematicus, which can be found in 40% of the articles. Other species that was being targeted Vibrio havi and also some species. This is not surprising for the Vibrio parahematicus to be the highest selected targeted bacteria due to its abundance in the aquaculture environment and also aquaculture organism. For the studies that did not target any bacteria, common genera found in their studies are Escherichia species, Acinetobacter, and also Pseudomonas. There were about 68 types of antibiotics identified, which were tested in these studies, with most of the studies tested ampicillin, tetracycline, chloramphenicol, and also gentamicin. From the study also found that ampicillin was found to have the highest incidence of resistance in bacterial strains. The aquaculture products and environment that are being studied by these papers where they look at water samples, sediment, or aquaculture products, with aquaculture products being the most focused, and the common sample aquaculture products being tested are shellfishes, which constitutes of mollusks and also crustaceans, and also some fish samples as well. Overall, we also found that the isolate samples from the water, sediment, and aquaculture products reported high prevalence of resistance towards ampicillin and also tetracycline. In conclusion, continuous monitoring and surveillance is needed in aquaculture products and aquaculture environment, especially on the resistant issue in ampicillin and also tetracycline antibiotic in Malaysia. Further understanding on the widespread of the antibiotic resistance in aquaculture products and its environment is essential to be studied in future research. Before I end with my presentation, I would like to acknowledge that this study is funded by Geran Galakan Pendidikan Universiti Kebangsaan Malaysia. And with that, thank you.